Okay.
Great.
Awesome. Great. So hello, everyone, and thanks for logging in today to hear about what I've been working on my past 5 years as a PhD student. And I'd like to especially thank my committee for being here. So this includes my advisor, Sasha and Minsun, and then committee members Anne Greenbaum and Mariam Fazl. So today I'll be talking about some different ways that applied mathematics and optimization in particular can be used to guide treatment planning for cancer therapy. Now, there's different types of cancer therapies used in current practice, but underlying all of them are the competing objectives to both maximize your tumor control and at the same time minimize your normal tissue complication. And being able to frame treatment goals as either a maximization or a minimization problem allows us to approach them using optimization methods. And radiation oncology in particular has a rich history of collaboration with mathematicians. So today I'll go over 3 projects that use optimization for different aspects of the cancer treatment planning workflow. I'll start with a brief overview of a project that uses dynamic programming for multimodality treatment planning. And this will be a higher level review because this was presented already in my general exam. And then next I'll cover 2 projects in radiotherapy treatment planning. First, we'll look at a nonconvex approach for Fluence Map Optimization that focuses on new results since my general exam. So this includes some new theoretical results and a new re-weighting strategy. And finally, I'll present a novel hyperparameter optimization approach for automated radiotherapy treatment planning that has the potential to both reduce the treatment planning time and improve treatment plan quality. And all of these projects have numerical examples. So I've made all of my project code and data available on GitHub. Okay, so let's jump into the first project. So like I said, there's many modalities currently used to treat cancer. Some of these include surgery, chemotherapy, radiotherapy, and most patients receive 2 or more modalities, often sequentially, in the course of managing their cancer. Now, in current practice, multimodality treatment decisions are often based on empirical intuition or clinical experience. But with so many different modalities and patient characteristics and possible outcomes, the optimality of these types of decisions is often unclear, and a heuristic search for optimal multimodality treatment policies can be costly and inefficient. There have been some limited efforts to approach this problem mathematically, most of them using a differential equations approach. But in this project, we propose a novel mathematical framework for these multimodality treatment planning policies that uses a finite horizon Markov decision process approach. And this model has the potential to be personalized based upon patient states and custom utility functions. And it can also be used to help explain treatment decisions to a patient. This project is published in the Journal of Mathematical Medicine and Biology, and you can find all of my code on GitHub and Zenodo. And this also includes an interactive GUI where you can change the utility functions and see how the policies change. So a Markov decision process, or an MDP, with a finite planning horizon, is a mathematical framework for optimizing a sequence of actions within a stochastic or random system, where the state of the system is given at the beginning of each decision epoch, and then the goal is to maximize the expected reward over the entire planning horizon. So in our setup, we have a treatment planner that observes the patient state at the beginning of each treatment period, and then they choose a treatment modality based upon the state that they observe. The patient state then transitions to the state in the next treatment period based on transition probabilities determined by the state in the action. And then after each treatment period, we can receive intermediate rewards based upon the patient state and the modality used. And then at the end of the planning horizon, we receive a terminal reward that quantifies the benefit of being in the final patient state. Now, since this system is stochastic, the ultimate goal is to calculate a policy or a sequence of actions that maximizes the expected reward over the entire treatment course. So for our model, we define 3 different types of treatment modalities based upon the risk of increasing side effect and the reward of decreasing tumor progression. The first type is classified as high risk and high reward, but it can only be used once during the course of treatment. And the second type is lower risk and lower reward, but it can be repeated. So these 2 types reflect the fact that certain modalities might be more effective, but limited in how many times you could use them. So for example, whole brain radiotherapy could only be used once during a patient's lifetime. So that would be classified as type 1, but gamma knife or partial brain radiotherapy can be used multiple times. So those would be classified as type 2. And then we include a third treatment type that's actually no treatment. And while this doesn't decrease your tumor progression, it allows for side effects to reduce. The patient state is then defined in terms of 3 state variables. The first one is a history variable that keeps track of whether or not you've used the type 1 modality. And then we have the degree of side effect and the degree of tumor progression. So in both of these cases, a lower number is better. So 0 means no side effect and tumor remission. And then the higher values are the worst possible states. So in our model, M or N means patient death due to either side effect or tumor progression. For a small state and action space, we can actually calculate the optimal policies exactly using the backward induction algorithm. And this approach starts at the final patient state and then works backwards, determining the action at time t-1 that will result in the highest expected reward for each state at time t. And here, VT is the value of being in a particular state at time t, and then A of t is the action that maximizes this value at a particular state at time t. So here's an example of an optimal treatment policy, which you could use to determine the best action for any given patient state and treatment period. So for example, if you are in treatment period 1 and you haven't used the type 1 modality yet, you would look in this first row and first column. And then you can look up your optimal modality looking at the tumor progression on the horizontal axis and the side effect state on the vertical axis. So overall, our policies align with the clinical intuition that if you have higher tumor progression and lower side effect, you can afford to use the more aggressive modalities, versus if you have lower tumor progression and higher side effect, that's when you want to be a little bit more conservative. So in our paper, we included additional examples that demonstrated that changes to both the reward functions and the transition probabilities resulted in intuitive changes to the optimal policies. So to summarize this project, we looked at the problem of that the optimality of multimodality treatment decisions based upon intuition is often unclear. So we developed a mathematical framework for multimodality treatment planning that used a finite horizon Markov decision process approach. And then we demonstrated with our numerical examples that the policies align with clinical intuition. Now, this approach can be extended by estimating the state transition probabilities from data, using a larger state space, so including more additional relevant state variables. But this problem suffers from the curse of dimensionality. So as you add a larger state space or action space, the problem becomes infeasible in terms of solving it exactly. So in those cases, we would need to look at approximate dynamic programming methods rather than backward induction. And then another option for extending this is to look at a infinite horizon MDP rather than finite horizon. And that would be relevant in the setting where you're not sure how many treatment periods a patient will need. Okay, so that looked at choosing amongst different modalities. Now we're going to move to the setting of a specific modality, at radiotherapy. So radiotherapy works by exploiting the weakened DNA damage response of tumor cells. So specifically, radiation damages cell DNA, and because tumor cells proliferate more rapidly than healthy cells, they have less time to repair DNA damage, and the unrepaired damage can be fatal to the cells. Now, The most popular type of radiotherapy is external beam radiotherapy. Here we have beams of radiation delivered to the patient from multiple angles. And by using a combination of these beams, we can deliver a high dose of radiation to the tumor while limiting the amount of radiation that's delivered to normal tissues and critical structures. Each beam is actually shaped by something called a multi-leaf collimator. So here, the sliding metal leaves of the collimator can be used to shape the outline of the beams to match the tumor geometry. And then you can also modulate the intensity of the beam. So you can have higher intensities in some regions and lower intensities in others. And then this allows you just a greater control of the dose delivered to the patient, but it often results in a higher dimensional problem. So here's an example of intensity-modulated radiotherapy, where we can see that the beam outlines have been shaped by the multi-leaf collimator to match the tumor geometry. And then the beams have been modulated. So we have, in some regions, we have higher fluence than others. And this allows us to deliver a higher dose to the tumor in the center, but then a lower dose to the surrounding normal tissue. So in the early days of radiotherapy, the beams were much simpler shapes, more like rectangles and wedges. And then the intensity was uniform across the beam. So the way that treatment planning would work is the planners would just experiment with trying different beam angles and intensities until they found an acceptable dose. So essentially they were looking at the beams first and the dose second. But with intensity modulated radiotherapy, there's far too many variables to be able to do this trial and error forward planning approach. So what's done in current practice is known as inverse planning. So here we actually specify the dose first and calculate the beam second. So first the patient goes CT imaging. So we have a three-dimensional model where we know the location of the tumors and the different critical structures. Then the physician specifies the dose prescriptions for the tumor and the dose constraints for the various organs at risk. And finally, the treatment planner calculates the number of beams and their orientations, the beamlet intensities that deliver this prescribed dose to the patient, the collimator leaf sequencing that actually achieves these beamlet intensities, and then finally, how the dose is delivered over multiple treatment periods in a process known as dose fractionation. And each of these pieces in step 3 can all be formulated as an optimization problem, and they can be solved either in combination or separately. But we're going to consider specifically this problem of solving for the beamlet intensities, which is known as the Fluence Map Optimization problem. So in our problem setup, the beams are all discretized into units called beamlets. And then the body is discretized into volume units called voxels, which are labeled based upon which structure a voxel belongs to. And then we have some sort of mapping that goes from the beamlet intensity in a given beamlet to the dose deposited in any given voxel. And then our goal is to determine the set of beamlets that deliver the dose that matches the prescription. So here's an example of how we can formulate this problem, the Fluence Map Optimization problem, because we're optimizing the beam fluence or intensities. We often try to deliver a uniform dose to the tumor while satisfying various constraints on the organs at risk. And this comes from the fact that tolerable doses and constraint types are known empirically for different types of tissues. So some examples of this are serial structures such as the spinal cord and brainstem lose functionality if any of their subvolumes are damaged. And so in this case, maximum dose constraints are often used. On the other hand, parallel structures such as the liver and the lungs are composed of semi-independent units. So a fraction of those organs can be damaged without loss of functionality. So for this setting, we can apply a maximum dose constraint on a fraction of the organ. And this is known as a dose volume constraint. Now, while these first three constraints we have here are convex, the last constraint is non-convex. In particular, the dose volume constraint doesn't actually specify which fraction of the parallel organ can exceed the dose and which can't. So it's a combinatorial problem. So why does this matter? So convex optimization problems are really well understood. We have a lot of theoretical results, state-of-the-art approaches, and commercial implementations. But non-convex problems are a little bit more challenging. And we'll demonstrate one of these challenges with the toy problem. So here we're looking at delivering a uniform dose of 81 gray to one prostate voxel, while satisfying the dose volume constraint that at most 50% of the rectum can exceed more than 20 gray. And here we're looking at two voxels in the rectum. So that means one voxel can exceed 20 gray and one cannot. And then we look at the problem with just two beamlets. So we're able to actually plot the feasible space. Okay, so on the left, we see the dose delivered by our two beamlets, X1 and X2. And here the tumor voxel U receives about 81 gray. Voxel 1 of the rectum receives more than 20 gray, and voxel 2 receives 20 gray. And this is represented by the point B on the right. So this is contours of the objective function and then the feasible space for the beamlets. So this is a non-convex region where the bottom region is where voxel 1 is allowed to exceed 20 gray. And then the right-hand side is where voxel 2 is allowed to exceed 20 gray. And this problem has two local minima. The global minimum is at point B. And depending upon your initialization and the algorithm you choose to solve this problem, you could arrive at either of these 2 points. And there's really no guarantee that you'll arrive at the best solution at B. Now, in the case of a convex problem, any local minimum is also a global minimum. But in a non-convex problem like this, we might have many local minima, and there's no guarantee of finding the best solution. Our problem in particular is the feasible region is made up of the union of convex regions, each corresponding to a different choice of the subvolumes of an organ allowed to exceed a dose or not exceed a dose. So historically, this problem of local minimum has motivated the use of stochastic algorithms like simulated annealing to solve this problem. And these are able to escape local minima, but they tend to be inefficient. On the other hand, you can approximate the original problem as a convex problem, and then you can use more efficient methods, but you've lost the structure of the original problem. So our goal here was to develop a new formulation and algorithm for this Fluence Map Optimization problem with the non-convex dose volume constraints. That both preserves the non-convexity of the original problem, but at the same time can be solved efficiently. I'm going to focus on some new results since the general exam, including expanded convergence analysis and a new re-weighting strategy to improve solutions. This project is currently submitted, but in the meantime, you can find it on the archive. And then I posted a link to all of my code. So let's see, in the original problem here, a couple of difficulties were that one, the variable we're solving for the beamlets X appear both in the objective and in this non-convex constraint. And this constraint, this set defined by the constraint is difficult to project onto, partially because of this linear map here. So our model instead uses a auxiliary variable W, and then we require that the dose to the OAR doesn't have to satisfy the constraint exactly, but it has to be close to a variable that does satisfy the constraint. So this is slightly easier than the original problem, because now this set is much easier to project onto. And then if you look at the function in terms of x for fixed w, this is actually just a non-negative least squares problem, which is convex. So to solve our problem for both x and w, we first view it as a value function optimization problem. So here we're projecting x out of our objective f, and this gives you the value function g. And this function is both differentiable and Lipschitz smooth. And so one natural algorithm to use for it is projected gradient descent. Here's like a high level of our problem or our algorithm that we're using. So we can have multiple organs at risk that have multiple dose volume constraints, and each of these constraints has its own subvector w that makes up our entire vector w. And each iteration, we take a gradient step in the subvector and then project it on its part of the feasible space. And so it's a little bit different from typical non-convex projected gradient descent, where we know that non-convex projected gradient descent converges if the step size is less than 1 over the Lipschitz constant of the gradient. The only problem is that our algorithm updates each subvector separately, and each can have their own step size. So we've expanded our convergence analysis to include this case. And here's a more detailed view of our algorithm, where each iteration we're solving a subproblem for x, and here we use a sparse non-negative least squares solver. And then each subvector is updated with a gradient step where the gradient is computed using our solution for x. And then our stopping conditions involve the difference between the subsequent iterates, which is scaled by the step sizes. Okay, so since the general exam, we've expanded our convergence analysis. So this includes a variety of lemmas, a couple of theorems. So the first important result is that the value function g is strongly convex. So even though our problem is non-convex, this function is strongly convex. And one nice thing about a strongly convex function is that not only are the local minima also global minimum, but we actually have a unique global minimum. And we can use the strong convexity of this value function and the specific structure of our feasible set to prove that the fixed points of our algorithm are all local minimizers of our value function on our set. And furthermore, if each step size is less than or equal to 1 over the Lipschitz constant of the gradients, we can prove the convergence of our objective value. But unfortunately, this only involves subsequence convergence of our vectors w. So now if we have a slightly stronger assumptions that our step sizes are strictly less than 1 over the Lipschitz constants of the gradients, now we can prove the convergence of our vectors w, and we can derive a convergence rate of 1 over epsilon. So one question would be, well, if you have better results for the stricter step sizes, why even bother letting the step size equal this value? And the answer is that in that case, the algorithm actually reduces to block coordinate descent, where we're alternating between optimizing for x for a fixed w and then optimizing for w for a fixed x. And one thing that's nice about this is we no longer have the hyperparameter of the step size to worry about. Okay, so here's a bigger example from our toy example. We still have the same uniform dose target of 81 gray for the prostate, but now we have the dose volume constraint for the rectum that at most 30% of the volume can exceed 30 gray. This time we have 986 beamlets, and then the size of this variable w corresponds to the size of the rectum, so about 1700 voxels. This is a solution using the block coordinate descent algorithm. So let's see. So we have about 81 gray delivered to the tumor And in this cross section, very little of the rectum receives more than 30 gray. And then here are the different beamlets. So this is actually the X vector. And we can see that the shape of the beams correspond to the shape of the tumor from the different angles. And then we have some regions of higher intensity where the beams only pass through the prostate. And then we have lower intensity regions in parts of the beam that also pass through the rectum. Here's a plot of the convergence of our objective value. And it's interesting to look separately at both the PTV term and the OAR term. So our initializations all use the solution where we ignore the dose volume constraint. So this is where we have the best dose for the tumor, and that's where the tumor objective term is the lowest and the OAR term is the highest. But as we start iterating, we lower the dose to the OAR so that that term decreases, and then the tumor term has to adjust. So the dose to the tumor has to adjust to this competing objective. Here's a plot of the convergence criteria, so the difference between the subsequent W vectors. And then on the bottom, we're comparing how much of the rectum received more than 30 gray. So at our initialization, about 65% received more than 30 gray. So that's pretty far away from meeting our constraint. And then when we converge, a little under 35% meet the dose. And so we didn't actually meet the constraint. And this is one of the trade-offs of using a relaxation, that we're able to solve the relaxation more efficiently, but we're no longer, it's no longer a hard constraint, so we're no longer guaranteed our solution will meet the constraint. However, some things that we noticed when we were experimenting with different prescriptions was that if we solve the problem for a lower dose value or a lower percent value, our solution met the constraint for an easier problem. So essentially, the solution to a more difficult problem was able to satisfy the constraints for an easier problem. And this then motivated a re-weighting scheme where we iteratively solve more difficult problems in terms of increasing the weight to the OAR term and decreasing the dose and percent values for the dose volume constraint until we eventually solve the original problem. And because our algorithm is fairly efficient, we can afford to solve multiple problems until we find a solution that works for the original problem. So here's an example where we compare between the original relax problem and this re-weighting scheme. And we use two different stopping conditions for the re-weighting scheme. We compare on what's known as a dose volume histogram. So these are read by any point on this curve tells you the percent of an organ volume that receives at least a particular dose. So for the prostate, we'd ideally like 100% of the organ to receive exactly 81 gray. So we'd like it to look like a step function. For the rectum, we want it to meet the constraint that at most 30% receives more than 30 gray. So our goal is to have these curves travel through the box. So our initialization is this dashed line. And since this is the solution to the problem without a dose volume constraint, we don't really come close to meeting the constraint. The solid line is a solution to using the block coordinate descent method, where again, we've reduced the dose to the rectum, but we still haven't met the constraint. So next we use the re-weighting algorithm. The dotted line is where we solve the more and more difficult problems until we meet the constraint exactly, and then we stop. But one thing to notice is that we've changed the dose to the rectum without really changing the dose to the tumor at all. So one question would be, how low can we continue to decrease this dose before we no longer have an acceptable dose to the tumor? And so this dotted and dashed line here is where we've continued to decrease the dose values in the objective function until the dose to the tumor is 2% worse with respect to lower dose volume constraint called a D95. So here we can see that we've actually not only been able to meet the dose volume constraint, but we can actually reduce the dose below it by modifying the values in the objective function. And this is actually the motivation for the final project. Okay, so to summarize this project, we looked at the problem of Fluence Map Optimization for intensity-modulated radiotherapy, where the clinically relevant dose volume constraints are non-convex. So they're a little bit more difficult to solve. We developed a new formulation that can be solved efficiently, while at the same time preserving the non-convex structure of the original problem. And we proved that our algorithm, which is based on projected gradient descent, converges to a local minimum of the problem. Now, to extend this project, we can apply the model to additional tumor types and different patient geometries. We can incorporate the other constraints for the problem, and then we can compare our results to the performance of commercial treatment planning software. Which brings us to my final project. So this is my most recent project, and I got to use the clinical treatment planning software that they use at UW Medical Center. So this is the RayStation treatment planning system. And all of our examples for this project were performed on patients with lung tumors that were treated with what's called stereotactic body radiotherapy. Now, the treatment planning software is solving the same problem that we just looked at, the Fluence Map Optimization problem. But in this case, we're taking a step back and looking at it at a bit more higher level, because we don't necessarily know the form of the objective function that the planner is using. So what the treatment planner can do is they take the prescription and then they get to assign, they can choose different objective function terms corresponding to the different clinical goal types. And then they can specify dose and percent values, which I'm calling theta, and they can assign weights for each term. And these weights can be used to specify the relative importance of meeting some goals versus meeting others. Now, just like my other project, these objectives often use relaxations instead of hard constraints. So the treatment plan that minimizes a given objective function isn't guaranteed to be clinically acceptable. So to find a suitable plan, oftentimes the treatment planner has to manually tune the objective function. So manually tuning the objective function parameters and the weights in an inefficient trial and error process. And one problem is that the treatment plan quality is often determined both by the amount of time the treatment planner has to do this manual tuning process, but then it can also depend upon the actual skill of the individual planner. So treatment plan quality can vary depending upon these factors. So to improve the efficiency and the plan quality, there have been various approaches to either guide or automate this manual planning step. One popular approach is called knowledge-based planning, and this uses a library of previously optimized plans to predict both the dose values and the weight values that might work for a new patient. And while this approach can reduce the planning time by identifying a good starting point, it still requires the planner to do this manual trial and error step. And it also requires that you actually have access to a library of previously optimized plans that are similar to your patient. And sometimes that data is just not available. So our approach was to try to automate this trial and error step by viewing the problem as a hyperparameter optimization problem. And then we can use methods for hyperparameter optimization from the machine learning community. So this project is still in preparation. In the meantime, you can find my code on GitHub and Zenodo, and then we should have something on the archive soon. Oops. Okay. So in machine learning, a hyperparameter can include values either related to the structure of a learning model, so the regularization weights or the number of layers in a neural network. Or hyperparameters could also be related to the optimization approach used to train the model. So for example, a learning rate. And then the goal is to minimize a loss function with respect to the hyperparameters. For our problem, for the radiotherapy problem, we want to maximize a treatment plan utility with respect to these weights and dose parameters that RayStation is using in its objective function. And then our utility, which is using the results of the solution from RayStation, is defined so that we can quantitatively compare different treatment plans to each other. So we can assign a number that says this treatment plan is better than this treatment plan according to clinical intuition. And in our utility, we assign a different utility term to correspond to the different clinical goals that we have in our prescription. And each of these has a prescription value that we're calling gamma. In our case, it corresponds to the dose, in the doses in the prescriptions. And while we're specifically using the RayStation system, this model and this approach could be used for any treatment planning system that has a scripting interface. So you can interact with, for example, Python and the treatment planning system. And we don't require access to any other previously optimized plans. And then we show that we can actually improve upon manual plans by exploring a larger parameter space than what is typically considered in clinical practice. Okay, so that's our formula. Let's talk about our approach. We face a couple of challenges due to the fact that our utility is evaluated by solving a problem in RayStation. So this is a black box optimization function since we have no closed form function. We don't know anything about the convexity, the smoothness, differentiability of our function. Second, it's relatively expensive to evaluate because each time we compute our utility G, we have to solve this subproblem in RayStation. And depending upon the difficulty of the problem, it can take between 2 to 5 minutes for RayStation to calculate the beamlets. And while this isn't as expensive as like training a neural network, we have to keep in mind that the treatment planner has a limited amount of time to compute treatment plans for each of their patients. A final challenge is that when we compute our utility, the values we get can be noisy. So this uncertainty can come from a couple of different sources, including the solver settings in RayStation. So for example, you could hit the maximum number of iterations before you've actually converged. Another source of uncertainty is the dose calculation algorithm that RayStation is using to actually calculate from the beamlets to figure out what the doses are to each region. So one naive approach for hyperparameter optimization is a grid search. And this is where you sample uniformly from the parameter space. But this suffers from the curse of dimensionality, and it's not feasible for any reasonable size problem. So two other approaches sample in a more smarter or efficient way. One approach is to use a model-free method like random sampling. And one benefit to this is that you can run it in parallel. And then another approach is a model-based method like Bayesian optimization. At a high level, this approach iteratively builds an approximation to the true utility that we don't know. And then it optimizes this approximation, which is hopefully more efficient to evaluate. All of our examples, we used a random sampling method and a Bayesian sampling method from the Python package scikit-optimize. And this approach, for the Bayesian approach, it starts with a prior over the utility landscape, and then it iteratively samples from the utility function by solving a problem in RayStation, and then it refines the surrogate using a Bayesian posterior update to figure out where to sample from the parameter space. Each iteration, it optimizes an acquisition function, and this acquisition function tries to quantify the benefit of sampling from different locations. So these functions are designed so that their optimal points reflect the two objectives of both exploring the parameter space, so sampling where the surrogate utility has high uncertainty, so we get a better approximate function. But then at the same time, we want to exploit where we think that we have high utility, because ultimately our goal is to maximize our utility function. So in the function we use, GP minimize, the surrogate model that we're using is a Gaussian process. This describes a distribution over different possible functions. And for any set of weights and parameters, we have an associated random variable for the utility, which is modeled as a Gaussian distribution with a mean and a standard deviation. So for all of our patients that we compared, they all have the same 11 clinical goals that we are trying to meet. And then we have 10 tunable parameters. Our examples, we've set all of the weights to 1. And then we have normalized all of our plans to meet this goal, which is the D95 goal for the tumor. So for the rest of the goals, these values in orange are the prescription values, which we're calling gamma. And then we tune our dose values in the objective function for RayStation over the range from 0 up to gamma. And then each utility, each goal has a corresponding term in the utility function. And we use a combination of linear and linear quadratic utilities, depending upon how important each clinical goal is. So for some of our goals, there is a degree of flexibility in that we can violate them if it means that we can meet the more important goals. And for these, for the chest wall and the ribs, we use the linear utility, which can be interpreted as the percent difference from the prescription value. So if we are below the prescription value, we have higher utility. And if we're above the prescription value, we have lower utility. But one thing to note is that this sort of utility doesn't distinguish between whether or not we've actually met the goal in terms of just like the amount of reward that you would get. So for the more important goals, we use a linear quadratic utility in orange. So here our utility increases linearly if we've met the goal, but it decreases quadratically if we haven't met the goal. So this prioritizes getting the dose out of this bad region into the better region. So for most of the goals in our examples, we use the linear quadratic utility. Okay, so we wanted to compare the behavior of the two algorithms against some sort of ground truth. So for this case, I wanted to do a grid search. So I had to look at a toy problem where I only have 2 parameters. So here I'm setting all of my parameters at the prescription values except for those that correspond to the max dose to the ribs and to the, this is a 2-centimeter ring around the tumor. That's what this region of interest is. And now just to demonstrate how infeasible grid search is, to compute all of these treatment plans took me about 3 days. So you could not even dream of doing a grid search for this 10-dimensional problem. Okay, so what I'm plotting here is for each treatment plan computed with a given rib parameter and 2-centimeter parameter, I'm looking at the resulting maximum dose to this first organ and the rib. And this goal is actually pretty difficult to meet. So we really want to be in this region here to be able to meet the goal. Whereas most of the plans I computed did meet the RIB maximum dose goal, but this is the region that gives us the lowest doses. So ideally, we'd like to have a utility function that satisfies kind of the best of both worlds. So we'd like to both meet this goal while also getting a good dose for the RIB. So we'd like to have good utilities kind of in this region here. So here's a plot of the utility computed on this grid search. And I've truncated the color range so that we can see the region where we have positive utility. And it's nice because this corresponds sort of to the region where we meet this clinical goal. And the purple are the samples from the random sampling method, and then the green are the samples from the Bayesian sampling method, and then the stars are their optimal values. So they all achieve optimal values in a similar region, and then they all achieve similar objective function values where neither of the automated methods got quite as good as utility for the grid search. And then one nice thing about this Bayesian method is that while it's searching over the space, it's also building this surrogate model of our utility. So here we're plotting both the mean and the standard deviation of the surrogate model. And something that's cool is that this method's identified a similar region of positive utility to what we saw in the grid search. And so what happens is that the Bayesian method focuses most of its samples in this region of positive utility. So it avoids wasting samples where we're not going to get a good plan. Whereas the random method, it's sampling uniformly. So it does waste a lot of time in regions that we know won't be good plans. Okay, so a better utility only matters if it corresponds to better doses. So we have to look at what our resulting doses are. Here, for both the grid search, the random search, and the Bayesian search, we're looking at the dose values to each of our different goals. And we're expressing the dose as a % difference from the prescription value. So since these were all maximum dose type constraints, we want to have negative numbers. And we see that all three methods got pretty similar doses. The Bayesian method was the only one to actually meet all of the goals, but they're all very close. So this is promising, especially because here we've only been looking at 2 parameters, but our problem has 10 possible parameters to tune. So we can definitely do a lot better. So now we look at the 10-dimensional problem. So here now we're tuning all of the dose parameters for all of our clinical goals. And now we're comparing it to the plans that they actually used in the clinic. So the orange line is the utility from the clinical plan. The blue is the utility if I don't tune any of my parameters. I just leave them all the prescription values. And then green and purple are the utilities from the random sampling and the Bayesian sampling. Now, one thing with the Bayesian method is that we do some random samples before we build our initial surrogate model. So when I used 100 iterations, I let the first 20 be random samples. So in that case, my 2 automated methods were actually identical. But then after the 20 iterations, now you can see that by optimizing the surrogate function, the Bayesian method is able to get a little bit better utility than the random sampling. And now we look at our dose values. First, we look at them as a % difference from the prescription values. So again, the more negative, the better for this case. The clinical plan for this patient met all of the goals except for the maximum dose to this 2-centimeter ring, whereas both of our automated methods did meet all of our goals. And then in this bottom portion, we're comparing the dose to the automated plans with respect to the clinical plans. So we can see that our dose in the automated plans are lower than the clinical plan for all of our regions except for the PTV. So we've increased the maximum dose to the PTV in exchange for a lower dose to the spinal cord. And this is actually one of the more important goals. So this is a nice trade-off, especially since the PTV dose is still within the clinically acceptable range. So we compared our results for 5 different patients. Overall, the random search method was about half an hour faster than the Bayesian search method. But this made sense because the Bayesian method had some additional steps because we were both fitting a surrogate model and optimizing an acquisition function. And it seemed like a good trade-off because the Bayesian utility was on average about 5% higher than the random search method, and then about on average 40% higher than the clinical utility. Though in this case, one of the clinical plans had a particularly low utility, so it kind of skewed the mean. So the median was about 11% higher for the Bayesian method. For the first 4 cases, we actually found better plans using our automated method. In particular, the Bayesian plan delivered on average about 20% lower dose than the clinical plans, where the clinical plans violated the goal. And then in the cases where the Bayesian plan had a higher dose than the clinical plan, it was still lower than the prescription value by about 50%. So it was still within the clinically acceptable range. Now, the 5th case was a little bit trickier. Here, we ended up finding similar plan utility to the clinical plan, but we wouldn't say that they were better plans. Instead, they found a different trade-off amongst the different doses. So here, we ended up giving a little bit higher dose to the chest wall and the ribs, but a much lower dose to the spinal cord. And this makes sense because of how we designed our utility functions. So we gave both the chest wall and the ribs a linear utility. So in a sense, we're saying this is a little bit more flexible in terms of actually meeting the goal or not. Whereas the spinal cord had this linear quadratic utility. So it really emphasized getting out of this region where we don't meet the goal. So this kind of highlights the fact that you need to be careful about designing your utility functions because it's not easy to translate your clinical intentions into a mathematical function. Okay, so to summarize this project, we were looking at this, the problem of this trial and error process of manually tuning objective function weights and parameters that often results in a varying degree of treatment plan quality. And we showed that we could both reduce the treatment planning time and improve the quality using a hyperparameter optimization approach, both using random sampling and Bayesian sampling. And this is still an ongoing project. So some extensions we can do are to apply this model to different tumor types, to design more personalized utility functions, and to try to automate some sort of dimensionality reduction. One difficulty with the Bayesian optimization is that it can only handle up to a certain number of parameters before it performs about the same as random sampling. And so if we could automatically determine for a given patient which variables actually affect the utility and which ones could be set just at the prescription values, this might be able to improve the performance. And then all of our examples, I just use the default settings for the scikit-optimize functions. So we could possibly do better by either changing the surrogate model we're using or being more intentional about how we specify the kernel for the Gaussian process. And then we might also improve things by looking into the solver settings that we're using. So either the acquisition functions that are used or the methods that are used to optimize the acquisition functions. Okay, so let's summarize all of this. So within cancer treatment planning, we have kind of these competing objectives to both maximize our tumor control while at the same time minimize our normal tissue complication. And this makes it really natural to formulate a lot of problems in cancer treatment planning as an optimization problem. So we looked at 3 approaches to using optimization for cancer therapy. First, we looked at a Markov decision process approach to determine an optimal sequence of treatment modalities. And then we looked at a particular modality of radiotherapy, where we used a new convex relaxation for the Fluence Map Optimization problem. And then we looked at an automated approach for tuning objective function parameters in radiotherapy. Okay, so thanks everyone for your attention. I'd like to also thank the Department of Applied Mathematics, all of the staff, students, and professors for the fabulous experience I've had. And then I'd also like to thank the ARCS Foundation and the family of Dottie Simpson for their financial support, and also the eScience Institute at the UW for their financial support. So now I'd be happy to answer questions.
Great. Thanks very much, Kelsey. Maybe we'll start from questions from the audience before committee.
I'll ask a question.
Okay.
Of these sort of different projects, but with obviously a similar theme, has there been a particular project that has been most enjoyable or most interesting to you within the realm of the different things that you've covered?
Yeah, I think the 3rd project was my favorite for a couple of reasons. First, it's the one that I was kind of most involved in terms of formulating the problem. So I was more like in the driver's seat of noticing something about my second project, turning that into a new question. And then I got to use a treatment planning system. So that was pretty fun, trying to figure out how you actually interact with the treatment planning system, and then get to actually beat the clinical plans using my method. So I had a lot of fun with that project.
Very cool.
Oh, Obina, go ahead.
Oh, hi, great presentation. I was wondering, especially for the 3rd project you discussed, how receptive are the, I guess, medical practitioners that you work with to, I guess, the insights that, I mean, this research produces, or the improvements?
Yeah, so the problem I was looking at is like a known issue. So there have been other projects that look at trying to find a solution to kind of automate or guide this step. We haven't submitted it yet, so I don't know about the reception in like the wider community, but Min Sun could tell you, since she works in radiation oncology. She could tell you what she thinks of it.
I mean, obviously, I'm very excited about the last project that Kelsey actually led. And then I'm very impressed that, she came up with the idea and then she suggested, and then I was really, really kind of excited about the whole project, because right now the machine learning based automated treatment planning system is a very hot topic. However, you need a training cohort, so you have to have a bunch of plans, and then also the quality of the training cohort is very important. So that's why it's not yet clinically viable in practice, whereas Kelsey's approach is just a hyperparameter tuning, so you can utilize the existing treatment planning system with some kind of a scripting interface. So that is much kind of a feasible solution to current practice. And we just submitted the abstract to a conference this July, and then I'm actually kind of looking forward to it. But I'm biased.
Cool. And it's proprietary, is that right, Minsoon? What the system is actually optimizing.
Right, right. So nobody knows. It's really like a black box.
Cool. Yeah, that really stops a lot of development, because you can't change the underlying optimization. So it's a very cool idea to then get around that by optimizing the hyperparameter.
That's right.
Cool. Any other questions from the audience before we move to closed session? All right. Well, thanks very much again, Kelsey, for the presentation. So let's ask the audience, the general audience, to leave. Anyone on the graduate faculty can stay, and then we'll wait for that to happen and proceed to the grilling stage, as it were.
I think we can stop recording now too.
Oh, cool.